Good morning, everyone. My name is Katrina Chapman. Today is December 21st, 2020. Today I'm going to be discussing an article that I read in my Genetics Bio 1040 course. This article basically discussed the link between DNA structure and chromatin structure. The Genomic Code is basically encoded and modeled out of chromatin structure. And so this article was emphasizing how the Genomic Code solved the mystery of non-coding DNA. So then you might be asking, okay, what is this mystery of non-coding DNA? The article explains that non-coding DNA is basically DNA that does not code for proteins. So it makes up about 2% of the human genome, and it's been a mystery for the last 50 years. It's been termed junk DNA or selfish DNA because scientists didn't know exactly what the purpose of this DNA was. It wasn't fully understood. Now, non-coding DNA involves isochores and their role in the formation of chromatin domains and spatial compartments. So this article reviewed research that had to do specifically with those isochores and mapping of the isochores in those spaces and compartments. The recent study of these isochores led to an understanding of overlapping for the transcription of these non-coding DNA or non-coding RNA. There's long chains of non-coding RNA that are resulting from DNA. In the article, the author Bernardi discusses compositional genomes, which are based on short sequence designs of DNA, which led to the discussion of the isochores. Isochores are fundamental for genome organization. However, their basis for existence was pretty much unknown until recently. Throughout the studies highlighted in the article, they mentioned basically that there are two gene spaces. There is a GC-rich space, which is known as the genome core, and then there is a GC-poor area, which is known as the genome desert. These also correlate and correspond to open and closed chromatin. So the genome core is the open chromatin, and the genome desert is basically the closed chromatin. The open chromatin evolved and underwent a GC increase so that it could be thermodynamically stabilized. However, the closed or genome desert did not evolve and did not undergo these changes. So, because these isochores correspond to these different domains, they're related back to TADs and LADs. So TADs are topologically associated domains, and then LADs are lamina associated domains. So these isochores, the genome core and the genome desert, correspond to the TADs and the LADs. The study of spatial compartments further reinforced this correlation through the Hi-C approach, which revealed a compartment alpha and a compartment bravo. And then another method that was used was the FISH method, which is fluorescence in situ hybridization, which demonstrated the areas of the genome core versus the genome desert, and highlighted that the genome core correlates to the compartment A, which was studied in the Hi-C method, and then the genome desert correlates to the compartment bravo, or B, that was studied as well in the Hi-C method. So understanding that these two areas relate helped solve the non-coding DNA mystery. So for my opinion of how all this makes sense and how this mystery was solved, basically after reading the article, it reminds me a lot of what happens when you go fishing, and a lot of people have those depth finders when maybe they're in deeper lakes and they're trying to see the best spot to drop their hook for the fish or where the fish are congregating, and they have those depth finders that they can attach to the bottom of their boat. basically analyzes all of the peaks and valleys of the lake, essentially. So all the TADs and the LADs and the different domains of the genome remind me of these peaks and valleys within the ocean or within the lakes that these people are fishing in. So it's just lake mapping and same genome mapping. So it's just isolating these specific areas, the genome core and the genome desert, and highlighting exactly how these correlate and allow the DNA to operate and work. So yes, these isocores do not specifically code for protein. However, they are essential for the organization of chromatin within our DNA. Thank you for listening. And I just wanted to show my cue cards that I used before concluding. So there's card 1, card 2, and the backside's blank of all of these. Card 3, card 4, card 5, card 6, card 7, card 8, card 9, card 10, 11, and granted I have all these cards, but I tried my best not to use them, so. And then just reminding me of my analogy for fishing. Thank you for watching. Happy holidays.